that one session because of this. Now we were talking about the different parts of the membrane. Chapter 5 and even tonight's lab's all about membrane transport. And I want to just go ahead and just, we might have had this already, we'll see it in lab, or you've done it too. When we talk about things which get through the membrane, either that can simply diffuse, and we'll end up using that word, simply across, are things which basically cannot get in the membrane unless we use a protein transporter that we'll talk about. We have sort of had two types of lists, and we might have made this list, and we might not have, but we'll make it again. So when we talk about things that can penetrate, it's basically things which are also hydrophobic, because it's really this fat that's the blocker. So if you're small, nonpolar, or if you're a fat, so if you're a lipid, which of course include the steroid hormones. And what are the steroid hormones? Yes, okay? And there's some other ones, but that's the only three you have to know. Estrogen, testosterone, and cortisol. And so, because I'll just use that, I won't say steroid hormones necessarily. But they can freely diffuse, freely go through this barrier, okay? So that. How about the small gases? Again, these are nonpolar, right? They don't have any kind of partial charge either, and they can diffuse across. That's going to help us with the respiratory system. Other small molecule that we talk about in lab, that we actually do, you'll see tonight if you haven't seen it, another small molecule, urea, can also diffuse across. Okay? And that's in the main list of substances. Again, they're hydrophobic themselves, they have that property, and they can penetrate the cell. Most other things can't. And so non-penetrating ones are basically hydrophilic. They like water, but the fat is the big barrier. And so those are going to be charged and polar. And we talked about in Chapter 2 the different classes of molecules. We're going to talk about them again. There were proteins and lipids and carbohydrates and nucleic acids. Well, obviously lipids can pass through, but not only charged like ions can't pass through, so sodium and chloride, etc., they can't pass, but polar molecules such as proteins have some charge. Carbohydrates also have charge as well, or polarity to it. We can't, those will simply pass across. They're going to need help. These do, but these don't. So we'll show you in Chapter 5, mostly next week, of how we can get some of these things in. But basically, right now, it's just the fact of getting those two down. What can penetrate and what can't penetrate without, actually using some proteins we'll talk about. So charged polar. Again, carbohydrates, what's the main carbohydrate that's for our body that we keep talking about?
Glucose.
Glucose. And again, I might have said this already, and I'm going to repeat things a lot too. We've actually talked about a couple of proteins as well. Protein hormones. These are steroid hormones. Have we talked about any other hormones? So insulin and glucagon, which we're going to even see today again, review that for glucose metabolism or homeostasis, those are proteins. So they aren't going to be able to cross and do their action. They're going to sort of stay at the surface and communicate to it. So they cannot pass through. So these carbohydrates, and again, insulin, glucagon are examples of things which can't pass. Okay? All right. And you'll see in tonight's lab, and everybody else in the other lab saw we used this list tonight as well, or in the lab. But you'll see this list again. And Chapter 5 we'll see it as well. But tonight that's what we're looking at. So again, we talked a lot about phospholipids. We talked about cholesterol last time, and really talked about that it was limiting fluidity. And we did that. If we didn't talk about proteins, if I left off there, all I care about at this point is just two terms. Because there's tons of proteins we're going to talk about throughout the course and in Chapter 5. So I don't care if we have this and this. I care that you just know those names. Integral proteins pass all the way through the membrane. Peripheral sort of are on one side. Again, there's lipid anchored, all this other stuff, and if you read the book, you got a lot of other terminology. As she said, that won't be covered if I'm telling you, which I don't really lie on that. So all I care about right now is that you know those two terms. Integral proteins go all the way through. Peripheral sort of are just one side or the other.
One side
Or
of the
the
membrane or the other?
other, right. It's either on the extracellular side or the intracellular side, but it doesn't span the entire width. I put this picture up and again, some people don't like it, because I'm telling you it's not on the test, so why are we looking at it? But again, there are specialized regions, I just like the picture actually, that are a little different, that we're not going to go in specifically. But the idea is, there are these basically proteins that anchor to those sections. They're called lipid anchored. And so when you stain these, you see sort of the red in these kind of larger regions of the membrane and the proteins attached to that. And these are called lipid rafts, and I think they're just cool. But again, sorry, not on the test. So, but you know, I'm telling you. Okay, this helps us review too. We did protein structure in last chapter too. I want to remind us, this protein is not a big block. Okay, it's got, yes?
Oh, so I was just going to say, in the human body, you talk about cells a lot, and I know there are a lot of different types of cells. Do you mean in general, because there's kind of red blood cells and then there's just all other cells, right? So are you talking about any cell in the body?
Yeah, any cell in the body. All cells in the body are surrounded by the cell membrane. Now, each cell might have different amounts of cholesterol, and again, some differences in how much of these various proteins are these lipids. But we're talking about the general idea: phospholipid bilayer, proteins are in them exactly. Now, later when we do the red blood cells, we'll see that will be unusual that it won't have a nucleus versus all the other cells. But yeah, at this point, very generic, all cells. Proteins don't look like a big block. They have what? Primary structure. We're doing that. That's the sequence of amino acids. What is this little thing? Secondary structure, such as alpha helix, that you're going to remember. Tertiary structure is showing, again, the three-dimensional structure here. So when you look at this, you can see, again, you have this hydrophilic, okay, I guess I was telling you that too, but kind of showing you the three-dimensional shape of an integral protein. And again, we can kind of see that on this side, this is in the cytoplasm, we expect these things to like water, those amino acids, to be maybe charged ones because they are hydrophilic, they like water. The ones here within that fat, the lipid, are probably going to be what? Hydrophilic or hydrophobic? Hydrophobic, like CH2, CH3. So when we're making this and folding it, that's sort of the idea. As far as carbohydrates, the fourth component, basically the idea is I'm not going to talk about it. It assists in function because I don't have time. Okay? So the main thing to remember from this is the properties of the phospholipid bilayer and a couple facts about cholesterol, like fluidity. Everything else is just background. Now, I'm not going to put a question on the fluid mosaic membrane model because it's too general bio for me. But basically, using what we just talked about, they kind of came up with the idea that this membrane is, it's kind of, you can read what it actually says. That's called the fluid mosaic. The real key with this is, I'm not going to make you cook this like a tongue that day. But the basic idea of this is, let me kind of go, if I didn't say this before, will be the following. Let me use the other picture. Sorry, I'll use this picture. The fluidity is important. I think I did talk about that. The idea that these proteins, they don't pass this way. Things can't pass through, but they can move along this phospholipid layer, and that's going to be important for signaling and things, because if we have a hormone out here that wants to bind to these proteins, that will receive it, we want these things to move in one place, to concentrate. And so these proteins can move across the membrane. So that's the idea of fluidity.
Is that the only direction, left and right?
Yeah, and that's the idea, that you don't generally move, across the membrane except these guys, right? So these proteins generally are only going to be sliding, laterally, through the phospholipid. That's why it's sort of it is a liquid-like, basically. Mm-hmm. Yes?
And you said the main point of carbohydrates is to just make stuff work?
Yeah, it's to modify function. In other words, you can see that's like, we could do 2 lectures on how it does that for the immune cells and stuff like that, and I'm simply choosing not to. Yes?
So the penetrating things or solutes can go anywhere, but
Wait, the protein is specifically for the penetrating, or?
Well, it will be, actually, we haven't done it yet. So the idea is, yeah, the penetrating one, go over, they can freely diffuse inside and outside of cells, right? They don't need help from other proteins. We're going to see these things are going to use, and this we'll see in chapter 5, in order for these things to get in and out, they're going to use some of these proteins to carry them in and out. So, we'll see there's a lot of different, proteins that are on the cell to do things. But the ones we'll do in chapter 5 will literally be showing you how we can use these proteins to either signal or else, carry sodium in or carry the protein in or carbohydrate like glucose, into the cell and out.
That was kind of how you explained with negative feedback, how the insulin has to bind to a receptor in order to get into the cell.
Right, and again, we'll sort of look at that again, but yeah, it's sort of the idea that we have glucose, which can't penetrate, and again, we'll deal a little with this next time, on next Wednesday, but insulin can't penetrate. It's going to bind to one of these proteins, which we'll end up calling a receptor, but it binds to the protein. It's going to signal this other protein to carry glucose in. And then we have glucose and we get energy, which we're going to talk about next in chapter 4, and be happy, happy, happy. So glucose can't just go in, and this is the idea of how we're controlling things. And then, of course, when we have the proper amount, insulin will go down, and we're not going to suck all that glucose in. We want to maintain it. But this is sort of the idea that a lot of things can't pass through, like all these protein hormones, but we're going to signal what happens through this protein receptor, through one of these. So all kinds of proteins are here doing functions. Chapter 5, we'll actually talk about these proteins here that are actually carrying these things in and out. And yeah, we'll deal with all of that in chapter 5, which will be a lot next week. But again, try to know the first 4 and we'll be good. And so that's the end of that.
The first 4 what?
First 4 chapters.
Oh, okay.
Okay, I had to think first 4 ones
I
too.
thought you meant protein.
Okay, 2 more things, 2 more parts of this chapter. Again, this whole chapter is cell biology and a little tissue biology. So far, all we've really talked about cells is the properties of the cell membrane, which is very important, which we're going to be talking about in chapter 5, etc. But we do want to, and I'm going to get to that right now, right? Yeah. We do want to talk about, and I'll put this up again, we are going to, without going into the details of general bio of what specifically all these things do, we are going to basically go through and talk about sort of, what each organelle, each part within a cell actually does. And so I'm going to go over that relatively rapidly, except emphasize certain points, only because, again, we expect that you've had general bio. That's sort of a prerequisite. And in general bio, you talk about this is what the mitochondria does, this is what lysosomes do. And so, but you'll see the language, you can just study it again. Some of that's note cardish type of thing too. But I'll emphasize certain things. So we're going to do that. That's the second part of this chapter, where we're doing right now. But, and I might have already said this in the first lecture, I want to re-emphasize it. I think I did. Again, I have to admit, I get a little confused. I teach like this class, 2 different other schools, and we're at different paces. I'm like, and I'm getting really old. So I'm like, did I say that? Okay, but structure matches function. Did I put this picture up the first day or not, out of curiosity?
No, but you said that.
But I said it. And so this is just, again, so it's not a test question, well, how big is the egg cell? But again, just emphasizing that although we show cells like this, okay, with all the organelles, different cells, these are all single cells, based on their function, look completely different. So the nervous system, a neural cell, which its function is to gather information and transmit it long distance, boom. There's the nucleus and all these organelles will be here. It has extensions to receive information all over the body and then something to send it. So it's a specialized cell. Structure matching function. The other example I love to use is the egg and the sperm, ovum and sperm. 2 cells, okay, both involved in reproduction. Carrying on the species, all that weird stuff. But do they look the same? Completely different function. They both have DNA and a nucleus. The egg cell contains a lot of cytoplasm. And within that cytoplasm are the messages that control development. Actually, so when the sperm finally gets there and you have that zygote, how do we control cell division and the development of certain, structures that were part of it? It's actually, and we don't even completely understand it, but it's basically messages contained within the egg. Women are in control right from the beginning. Men are simple, as illustrated here. Men's job is, there's my DNA. I need to get there. I have a tail. They literally get rid of most of the cytoplasm, okay? Most of the components of the cytoplasm. They don't need that. Their job is literally, we've got to get to that egg, and we are as simple as demonstrates, I think. Women are more complicated than men, and women are, no matter what we think, are in control. I've been married a long time. All right, but again, and please don't sue me for sexual harassment. I'm taking this lightly. I'm like, what? Give me a break. I mean, I'd be careful now. I got recordings, and so. All right. Okay, so all that illustrates. Now, you look at the book, and it's got all. This is an example too. I'm not going to be talking about vaults and all this stuff, But what I am going to talk about are some of these organelles. I like the idea of just the term organelles. Tiny organs. The idea we talk about membranes helping to concentrate things. We're going to show you lysosomes have enzymes that degrade things. We don't want them mixed with our DNA, so we need a nucleus to actually contain the DNA. We're going to put things in compartments. You look at the other tables in the book. They're going to list each one of these and talk about their function. Do not memorize tables unless you like to for fun. But I am going to go through what I want you to know from several of these in the next part of the lecture here. So I'm going to talk about the cytoskeleton because I don't talk about that in general bio that much. And you can see some of the functions. And we'll show some examples. When cells aren't all globs, right? They have structure. It's going to be like our skeleton. It's going to provide them shape, basically organize things. And we'll see when we get to tissues, it's going to be involved in tissues. Oh, by the way, too, because I don't even know on this one, just remember, by the way, on these handouts, a lot of times, the handouts sort of correspond, and all of a sudden people are looking, because I don't remember what. That sometimes there are things in the PowerPoint that I just don't put in the handouts, that you can always go back to the PowerPoints. And of course, there's some things which I only put on the board. But sometimes, because I can never remember exactly what I skipped, I don't know if this is in the handout.
It's not.
But, good. That's why when I started hearing paper rustling, I'm like, wait a minute, I should tell them. Where is this? But it is in the PowerPoint, and you can kind of look at it. Now, this table you don't memorize either, but I do want us to associate some proteins with these three types of cytoskeletal elements. Small to big. There's microfilaments, intermediate filaments, and the largest ones are microtubules. Now, what I want you to associate with these, and especially this will come into play, is that I want you to associate actin and myosin, microfilaments, intermediate filaments. Because and some of you even know already, when we're going to talk about skeletal muscle, or really all muscle contraction, it's using actin and myosin kind of going up against each other, which will come in the next unit. So go ahead and just kind of get that these are cytoplasmic elements here. These are proteins.
So, on the test, would you ask what type of protein? Name a protein that's a microfilament.
No. No. Because I don't have a lot of fill-ins like that. I just don't like them. But it's. For this one, it'd be hard to. Well, let's wait a few minutes and we'll have a closer to it. But I don't like. There might be some later in the course where there might be a couple like that. But overall, I don't like to have sort of. They're almost like too matching, too simple to me to do that. But I do want you to know sort of those. Now, this will come into play in a minute. This will be more like it. Because when we talk about microtubules, the biggest ones, they're going to be movement. And so cilia, and we're going to be talking about ciliated epithelium when we do that tissue in a few minutes. Flagella, we're on the sperm. And when we start talking tissues, then we're going to see the importance of this. And so I'm going to be talking about like cilia moving and stuff like that. And then you can remember somewhere maybe there'll be the term microtubules, that it's a microtubule, it's not these other ones. but I can't give quite all the questions like that. But I'm trying to get you closer to what I'm going to be testing on. So, but right now, associate actin with microfilaments, myosin with intermediate, and movement. And we're going to get to this next of like cilia, flagella, like on the sperm, with microtubules.
So you want us to associate microtubules with tubulin and movement of cilia?
Yes,
Okay.
but not sizes or any of these other ones that happen to be there. Quick pictures for fun. All these pictures are, I'm not gonna put a picture up. This would be microfilaments. But just so you look at a cell, we don't normally see how extensive the cytoskeleton is. If you stain them with different fluorescent markers, you can see that they're quite important within cells. Okay, they are the skeleton that can give it shape, and eventually we can show movement. So this picture is showing those for fun. So let's skip this. So I don't care that you know about centrioles. I don't care, I will take these out later, that you know sort of the structure of the microtubules within the cilia. So here's the one I want you to look at, not to memorize all this, but to illustrate the point. And the point was, it helps provide cell shape. A great example would be the microvilli, which are in the intestines to perform a function. Structure matches function. If the function here is to maximize absorption of food into the body, we want a lot of surface area. How are these things maintained? Why are they up there? If you look at this, you see that we use all of the elements of the cytoskeleton so that these will project up and provide that surface area. So just an example of showing you how we use the cytoskeleton to sort of maintain a certain shape that has a function. Okay? We increase surface area to increase the absorption of food, and the cytoskeleton helps us. We're never going to have a figure in which I have lines and you put what each one of these are. You can do that, I don't know, anatomy or whatever. Mitochondria. All you need to know, basically this. It's where, and really when it comes down to it, won't be that much. It gives food energy to ATP. We're going to talk a little about that today. It's called cellular respiration. For this course, that's all you need to know. We're not going to go into the details. Okay? The endoplasmic reticulum are all these membrane passages that make things. So here we want to sort of step back and look at this. We have two types. This and what they do. The smoothie R, we associate it with these three things. Bless you. I think that was a sneeze. This is where we make lipids. Okay? The smoothie R. Where's the smoothie R? Over here. We make lipids. This is the tissue which will detoxify, or the part of the cell that will detoxify poisons, okay, drugs. And we'll see in skeletal muscle, it actually stores an important ion, calcium. Okay? Those three functions associate with smoothie R, which you have here. Rough ER, which have these ribosomes that we're not going to really go into detail for this course. But the key is, in conjunction with this, basically, rough ER is where we make proteins. Because we talked about classes of molecules last chapter, and we talked about proteins and fats. And so we make them in different places. We make the fats, the lipids, and the smooth ER, and the proteins in the rough ER. And that's why the steroid hormones are made where? Okay.
In the fat.
Which are, what makes fat? Smooth ER. Okay. Insulin glucagon. What class of molecules are they? Proteins. Where are they made? Rough ER. So I'm just going to put RER. And you'll see this on the extra question. This is the idea of what I want you to start figuring out as far as my questions are going to be. They're not going to be a simple, rough ER, let's match it to make proteins. You're going to have to remember when you're thinking about things that insulin, you're going to know sort of how it works. Okay, we just even talked about it here. You're going to remember it's a protein. You're going to remember that it doesn't penetrate. It's hydrophilic. It's made in the rough ER. And instead of studying that per se in all these note cards, you have to just start knowing and associate that. So if you get a question in which I have, like, parts of this, and I'll have something wrong, I'll say, insulin is made in the rough ER. Insulin is a protein made in the rough ER that penetrates the cell. And there's part of that's wrong, okay? It's non-penetrating. But I'll have another part that will have all parts right. So to do those questions, you don't want to quickly go through this and you see one of it's right and like, oh, rough ER, yeah, that's a protein. And you've got to go basically, know this information. You can even write it down if you've been studying it, and then go through each question. Okay, that part's true, that part's true, that part's false, A is false. And you do that again. And then you'll find one, only one, that has it all right. That's why all these things you want to start just incorporating together, putting things together, instead of having totally isolated blocks. Because that is the property of insulin, and all these things are important. Okay? So, those are pretty big. Sorry, keep pressing the wrong button. Smooth ER, rough ER, boom.
Is urea and the gases made in the smooth ER too?
Well, the gases are basically gases that we're generally taking in and out of the atmosphere. Urea, okay, so those are chemicals. Those aren't made, I have to see actually where we make that. The liver makes it. It's actually part of a degrading process. That's where urea is going to be a waste product of sort of taking amino acids, the thing that have nitrogen, and breaking them down. They're actually going to sort of be, we'll show you probably where they are sort of made in a couple slides. Right now, all you have to know about the Golgi, and because I'll make sure I'll say this, will be when you look at sort of how these proteins are made when we do it, they're going to be, and again, just write this down, I'm not going to go into the details of this, they're going to basically receive these proteins, change them a bit, and then basically sort them. Are they going to be going out of the cell? Are they going to be going somewhere in the cell? Are they going in the cell membrane? So, no more details than that, just basically Golgi receives, modifies, and sorts proteins. So, not too much on that. Don't worry about peroxisome too much. Lysosomes basically digest things. So I'm not going to put this on for that. But when we look, because I want to make sure it's actually in these structures here. When we look at degrading things like amino acids and breaking things down, they're probably going to be, like urea will probably be made in the lysosomes. But I want to check for sure, and it won't be on the test. Maybe next semester, though. That's a good question. All right. But it breaks things down. That's all I care about. Nucleus contains DNA. End of story. Obviously not, but for us it is. I got bigger things to fry. Done with the cell parts. So, as you see, especially if you had general bio, we spent a lot more time talking about this. I've emphasized sort of not only what we want to know, but I've started to give you a clue of how you're going to want to know it. Okay? Tissues. We said this first day, a collection of cells that carry out a related function, and they must communicate with each other to function. As I said before, we won't worry about these terms. When you look at tissues, or it's not just the cells, it's sort of the matrix and the other parts that it basically secretes so that they can attach and form a tissue. But here's what we're going to do. We're going to summarize this on the board. We've seen some of this before, but here's how we're going to summarize. We have two more things in this chapter to do. Here's one of them. I'm going to draw sort of two cells here. Just generic cells. And again, you're not going to have to draw all this. But I'm going to talk about 5 different connections here. And we've talked about a couple of them already. And sort of, which one do I talk about first? Okay. So, I'm going to make them here. We'll make that. Make that. Okay? And if I'm doing it in this order, we'll call that 1. Just hang on a second because I want to be consistent. 2, 3. Now I'll go back. I'm going to put 5 things up here. 1, 2, 3. Where's the little thing here? 4, 5. These are basically connections of tissues. And we've talked about some before. That's like a smiling cell flying along here.
Looks like a whale.
Okay? So, you guys, at 2, you make fun of my art. I get it. Okay, that's okay. I think it's cool. Okay? I used to like to draw fish like this, and I'd have the swordfish and be going through another fish. And okay, anyway, sorry. The drug wore off. Okay, the blue, by the way, is going to be, well, I'll put this in a second. So let's do this. We've already had this somewhat, but I'm going to put it here. Now, this is like the 3rd time we've seen some of this stuff, so this might be important overall. So the first connection I'm going to talk about, again, or review, gap junctions. And really, this is a review. Gap junctions, you can see we have a direct cytoplasmic connection. You want to, again, remember connexin proteins. An example might be heart cells, cardiac cells, because it is the most rapid form of communication. I don't think I've said anything different here.
And it's the strongest.
Not the strongest. But it's the fastest. Number 2. Okay? And we're going to see this in chapter 5. I wish we could just come in Monday. I'd like to do it, but we can't. Anyway, tight junctions. Okay? You do, again, at this point, I'm almost just reviewing what you already have in your notes. You do want to remember those proteins that are listed, included in Claudin, to remember that. Tight junctions, these, again, all this is, you might even have to write it down again, but it prevents, movement of material between cells. Well, let me write this. I might say it differently than before. Movement of material between cellular compartments. Let me just write that. You're so far behind. Example might be, and I'm going to describe this again, the intestine or kidney. And we're going to see this one more time next week in chapter 5. So again, my illustration, and I'm just illustrating it again for this example, is let's say up here represents sort of the lumen of the intestine where all the food's coming in, and this side's the blood. Okay? If we're going to go to all the trouble, which we will next chapter, to transport all this glucose into the blood, do we want it sliding up back into the intestine? These tissues have a great deal of tight junctions. Okay? So this prevents movement between cellular compartments, okay? Basically going from one compartment to the other. And we'll see that again. And that's sort of what we need to know about. Number 3 is also a review. And then we do 2 ones I haven't talked about before. So number 3, again, were sort of the desmosomes. These are the strongest cell-cell connections.
Are these anchoring junctions?
Say that again.
These aren't anchoring junctions, are they? Desmosomes?
What do you call them?
Anchoring junctions?
Anchoring junctions?
Yeah, that's not.
It probably is. It probably is a class, but I'm not even going to use those terms. You can probably cross that out. Because I just didn't like that term. I just didn't take it out yet. It actually is, though. Strongest cell-cell junction or cell-cell communication. It is, you do want to associate desmosomes. Those are the protein. When do you need strong ones? An example are the cardiac cells. Okay? And so, again, later, I don't know if we even talk about it, not only do these connect each other, but they actually communicate to the nucleus to tell the, to stay put. But right now, we might not even talk about that. Really, that's all I care about that you know. Desmosomes, again, it is an anchoring junction. It's helped to anchor cells to each other. I just don't care that you know that term at that point. But, look at this. Okay, and in fact I should have, I'll basically have the language here for 4 and 5. Because before I didn't talk about matrix. Cells are basically joined or using matrix. Again, this is extracellular, outside the cell, matrix. You know, a bunch of different proteins. That's what's represented by this blue. And if you're in a tissue, you've got to attach to the matrix. Okay? You're not floating around unless you're a blood cell. And that is called the... Does everybody have Lynn's information that needed? Because I need the board. Number 4 is hemidesmosomes. Okay, now you have it. 4, hemi, you know, like half, so to speak, desmosomes. These are cell matrix connections. Okay? A lot of tissues have this. So when we're trying to anchor cells to the matrix, hemidesmosomes. We can put, it uses proteins called integrins. And we're done with 4.
Are you talking about a connection between the intracellular and extracellular?
No. I'm talking about a connection between, well, there'll probably be, you know, cytoskeleton. I'm talking about a connection between a cell and sort of this underlying stuff outside the cell. I don't know, is there like a, yeah, here. So, you have the idea that here's a cell, and they're connected to each other by the desmosomes, but they're connected to this underlying, and we've got, you know, sometimes we'll call it basal lamina. This has, you know, sugars and other proteins, collagen, things like that we secrete. In order to anchor it to that glue, we use this type of thing, hemidesmosome. So we're connecting the cell to the matrix.
So is the matrix just floating around in the middle?
Well, it's in these type of tissues, it's really not floating around. It's literally, it's almost like, you know, it almost, because of all these proteins, it's almost like a glue. You know, so it stays put, but again, it's stuff that we secrete outside, so it's made up of different things.
Oh,
It's like the mortar, you can almost think of it as the mortar between bricks, but it actually does a lot more.
okay.
Yes?
For desmosomes, you mentioned before also fastest communication between
No,
cells?
no.
Yeah.
The gap junctions are the most fastest, the rapid.
So strong is cell-to-cell junction, but I thought you mentioned like last week fastest communication.
If I said that there, I was wrong. But I mean, the gap junctions are the fastest because of the direct cytoplasm. Yes. Wait, say that again? Well, I guess the idea of what we're showing is, and even with if you look at this, with this, you know, it does, when it comes down to it, use proteins that are integral proteins, go all the way through and attach to the cytoskeleton most of the time. So it does do that. So it's really not just on, for these, just on the surface of the membrane. They are using these proteins, coherents, that actually pass all the way through and do connect inside the cell too. Last one. My little whale. When cells, and this, again, for us, it's, you know, it's basically, the immune cells are a great example. When cells have to crawl around, so this one's an immune cell trying to engulf a bacteria, you know, in order to crawl around on a matrix, okay, you know, they don't have like feet with motors per se. They basically have to do a process. And what they're going to do is basically sort of extend their cytoskeleton, which will be actin, and attach to the matrix here, kind of releasing back, slide forward. Extend their actin, attach, move forward. And where they're attaching, okay, are basically called focal adhesions. So, 3 things on this. We're talking focal adhesions. We're talking then the actual cytoskeletal protein actin, I want you to know, and it's involved in cellular movement along matrix, along this stuff that's outside the cells. Example being, of course, immune cells. Most of our cells in the body now don't do this. Most cells in our body pretty much stay put. But the best example of ones which involve a lot of movement on matrix would be these immune cells. Early in development, cells had to go all the way around to develop the nervous system, circuitry. There's a lot of that going on. We stay put, except for these guys. Of course, if you are a cancer cell, what do you decide to do?
Just replicate it.
We want to start metastasizing out. We actually redevelop, this is a side note, but I'm a cancer. We redevelop the ability to do this again. Okay, we used to be able to do it. It figures out what mutations. It starts to do that. That's how it crawls around and metastasizes to your different organs and makes things bad. Most of our adult cells say, no, we're going to stay put. Yes?
What do you mean by immune cells? Like
White blood cells.
Okay.
And again, this one is probably something involved in, which we'll do later, phagocytosis type thing where it involves. So yeah, white blood cells.
Okay.
So, those 5. 3 of them we've seen before, and 2 new ones here that we need to know. The hemidesmosomes, cell to matrix, using integrins, the focal adhesions, using actin, cellular movement across the matrix. Yes, ma'am?
So from what I'm understanding, like all of these cell functions is how they're reacting with one another, like to create these
Tissues.
tissues.
Except for this 5th one, we're talking about more of a movement thing.
Yeah.
But these 4 are, when we form tissues, and again, not all cells have all of them. Some have more of one than the other. And again, I want you to even realize that when we talk about those intestinal cells that transport, they have a lot of, compared to other tissues, tight junctions, right? If you look at cardiac cells, they have a lot of desmosomes and gap junctions compared to other tissues. Other tissues will have these, but the ones that have more of it will match their function. Okay? But all tissues, almost all tissues, have this extracellular matrix that they have to attach to, and they are using then hemidesmosomes. So yeah, all of that is sort of for almost all cells. It's just how much for each cell.
How come cardiac cells and heart cells just don't both fall under gap junction?
Because they have both. So, I mean,
Like a mix.
both of them exist at the same time. So when you're looking at the connection of heart cells, it's not like some have one and some have the other. They all are connected by gap junctions for their communication. But what's not shown maybe in this one slide, they're all connected by desmosomes for strength. And so when you learn anatomy, intercalated discs, if you would look at them, they have both of these. You'll see gap junctions and you will see desmosomes.
Okay.
Yes, sir.
So it's a kind of silly question, but this whole process is pretty much the reason why we don't just fall apart.
Yeah. That's how tissues, and again, what I'm not saying now, and we might not fully cover is, not only is it that, but what these things do is actually communicate all the way to the nucleus and actually direct the cells of what to do. And that's cell communication, that's chapter 6, we talk about how it does it, we might not get to it, but so it's not really just that we don't fall apart. It literally, let's say for skin cells, let's say I'm a skin cell, and I'm attached to these 2 guys by desmosomes. Don't I look like that? And my job is, I want to be a barrier, but that means I don't want to start dividing and going anywhere, right? And that actually tells the nucleus that. My job is to stay here, maybe secrete some keratin and basically stay attached. It literally tells it what to do. If you lose those connections, guess what's going to happen? That cell's going to go, sweet, I can do what I want. You know what I want to do? Multiply. Multiply. And what disease is that?
Cancer.
And again, I might not get to it that much next week. Maybe I'll do it for the next test. But cancer are mutations, most of them involve breaking these connections. And then you're breaking communication, and now you're free to do what you want. And usually the default pathway is divide. Okay, no more control. Divide, 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 and then you say, hey, what's the brain like? I've been in this thing 50 years. And then you develop these other mutations to start crawling around. So and again, all this is not, I'll write that down unless I cover it later. But the idea is that not only is this providing integrity to the tissue so it doesn't fall apart, it really is communicating to the cells of this is what you're supposed to do. Any questions about that? Last thing in this chapter. Then we take a couple minutes to breathe, and then we do at least a lot of a very cool chapter. So, tissues. So, again, you really just want to pay attention to this because everything you need to know are in these, your handout are in this, the PowerPoint. You know, there's not a lot of board stuff, so sort of just listen because it's sort of basic here. So, 4 tissues of the body, 4 basic types of tissue. Epithelial, connective, muscle, and neural. And you have this. Now, epithelial has a lot of functions, and we're going to talk about 5 different types. I've already sort of mentioned that, right? For function. I said that back in the first day. We're going to come back to that. But let's just look at the others. For connective tissue, which, you know, is like what? Bone, cartilage, blood. I think I have a list there. Overall, the big thing I want to associate with is just this, and then we don't talk about it at all in this for now. It doesn't have as many cells of these other tissues. It's mostly matrix. Okay? If you look at, you know, if you would look at the structure of bone, okay, you know, kind of make these, okay, don't make fun of this, okay, this is my bone, okay? You know, you have these passages, you know, made of these proteins and calcium, etc. You know, there's only a couple cells that make all this. So it's mostly matrix, not that many cells. And that's why I want you to associate with it. I don't want to go into more connective tissue than that, as far as details. Just realize it has a lot more matrix than cells compared to everything else. That's it. These other 2, again, I want you to remember there's 4 tissue types, but if you look at muscle and you look at neural, what do they both say? They can both generate and propagate electrical signals. What's our next unit? The nervous system and its control of skeletal muscle. So do we really care about this beyond this now? No. So that's why this is almost just know there's 4 types of tissues, what they are. And then realize the only thing you have to remember about connective tissues is this. The last thing, pretty straightforward, but this is important. I said first day that we were going to learn whether you learned these 5 types of epithelium by structure and anatomy or you didn't. Doesn't matter. Because all we care about are the 5 types of epithelial tissue, a great cartoon, that we're going to base, it's actually easier I think, we're going to base upon what it does. Does it protect? Does it exchange? Does it secrete? Etc. So here they are. So it's a very straightforward thing, and, well, you know, it's easy for me to say, I don't say you don't have to study it, but it's not going to be as complicated. Now, there is a table, and if you like tables, you can memorize tables, but do I like tables? No. But you can use it for your pleasure. So, but, and we can go back to the picture. The first one is exchange. Again, when we look at the alveoli and we look at most blood vessels, we want easy exchange. I don't like the word leaky, but in this case, you don't want tight junctions between cells, okay? And you don't want multiple layers. We want the gases and different nutrients to pretty easily go from one compartment to the other, and so most blood vessels, and you can kind of go back to the picture if you have color, most blood vessels, and not all the lungs, but the alveoli, are surrounded by basically exchange. Okay? That's what you need to know. These guys, we're going to see this again in Chapter 5. This is what I've been drawing. Okay? You look at transport of substances from the intestinal kidney into the blood. Chapter 5, we're going to go into many details of this. We looked at this. I put it up on the board a lot. Okay? They are usually connected by tight junctions, which you associate the protein. Claudin occludin. Yes, you have to study all that eventually, right? Tight junctions, claudin occludin, and it is in transporting epithelium. And the idea of this is, and we're going to go through the process, I talked about it, but we'll go through the details. If we want to get glucose out of the intestine into the blood, we're going to transport it through different proteins we'll see into the blood. Again, we didn't want things to go back, so we have tight junctions. We're going to talk more about this. But this is transporting epithelium.
The intestines is made up of most epithelial cells.
Say that again?
The intestine is made up of epithelial cells.
Yes, the lining of the intestines are made up of epithelial cells. Now specifically, this isn't actually everywhere when it comes down to it, but it's going to be in specific places where we do the exchange, which is just sort of in parts of the intestine. But yeah, basically the inside, the whole GI tract, just like the outside is epithelial, that's sort of the external environment, that's all epithelial type tissues as well. So, what next? Ciliated. So, the ciliated epithelium are basically, now, instead of being, a paramecium in which we move with cilia, in this case, these are the cells, but the cilia move. And cilia we associated with what cytoskeletal element? Microtubules. You'll study all this. But here's the purpose of where cilia is and what they do. We'll go back to this. There's generally two places in the human body we look at cilia. One is the upper respiratory tract, as things that are coming from the outside environment, gases, but we also have bacteria, particulate matter. Do we want them to get to that nice sensitive alveolar cells? We trap them, it's a mucus, and then the cilia moves the material up our respiratory tract to try to get rid of it. That's where one place we have ciliated epithelium. That again, the cilia are moving, using those microtubules, trying to move those particles and things up instead of down. That's where we have ciliated epithelium. The other one is the female reproductive tract. In this case, fallopian tubes, we want to move the egg down. And so ciliated epithelium here will, basically beat the egg down and down. Those are the two places for ciliated epithelium. Pretty much.
Why, how come it's not flagellated?
I don't know why, but just listen. Flagella is usually, really involved in cellular movement, okay? Versus this is sort of on the cell moving thing. And the only flagella we have is actually what? Sperm, okay? Because that needs to move through liquid, and different bacteria that have that are really moving through some sort of liquid environment. So that might, I don't know, it might be cool, like to have your lungs with all these things flying around, but I don't know. That's ciliated. The last type we're going to talk about, protective is protective. Not a lot to say about that. Is secretory. This is important. This secretes things. Now, I should put, on your handout you have both here. I have this on one page and I have a picture of it on the next. But the big thing with secretory epithelium is there are two types that we want to distinguish. Exocrine, endocrine. And again, here are the two bullet points and we'll show the picture. So exocrine are going to secrete things into the external environment using a duct. Endocrine into the blood without a duct. Again, you should have, these are directly on your handouts. And see, the picture of that is here. Exocrine is going to secrete to the external environment, and you can see this duct of where it's going to go. Endocrine goes directly in the blood. We've lost the ducts. If you look at, where's my picture? Go back. So if you look at this, I guess I want to make sure we sort of figure this out. When we think exocrine to the external environment with a duct, what do we think?
Hormones.
Well, not necessarily. What's external environment? Outside. The outside, the external environment, the outside. And so we usually think of what kind of glands? Like sweat glands. Okay, external environment. Sweat glands are an example of an exocrine secretion. But the more tricky one that I want to make sure we know is, if you look at the whole digestive tract, what's the digestive tract? Is that internal environment or external environment? External. Okay? It looks like it's internal because it's inside our body. The internal environment represents the blood compartment, okay, the fluid compartment. This is entirely external environment, okay, from top to bottom. Okay? It's in contact with the external environment, just like some of these are. But this is complete. So anything that we find that's going to be secreted for digestion represents exocrine. So saliva, exocrine. Bile, all the pancreatic secretions to digest that we're going to talk about later, but they come in, they're shown here. All those are exocrine. So some people won't get that because, again, they think, hey, this is inside me. They get the sweat, but they don't get that. Saliva, this is external environment. It's in direct contact. Yes?
So are you, bile a lot of times, in the stomach, isn't there a lot of things that the liver secretes into the stomach as well, and bile?
But, yes,
So I'm just wondering,
it's into the intestine, and that is exocrine.
Right, and that does that just through ducts as well?
Yes.
Okay.
Right, you know, it stores it in the gallbladder and goes through the bile duct, or whatever it's called, right?
Yeah.
So, and the same thing, what we're gonna see when we look at the pancreatic secretions, you know, the ones that go into the intestine, they're gonna be going through ducts as well. And so those are all exocrine secretions. Endocrine secretions go into the blood. Have we talked about any endocrine secretions, any endocrine hormones? Insulin, glucagon, steroid hormones. Okay, all of those basically are going into the blood. Okay, they're not going external environment. So insulin, glucagon, and even the steroid hormones all represent endocrine. Okay, by the way I'm talking, do you see sort of the hard part here? Okay, how we're talking about this and we're memorizing this, but we have to go back and remember this, okay? That these also are made, okay, all of these now represent endocrine secretions. And some people might ask, how are we supposed to know that? And you know my answer is? I just said it. Okay? Literally that. That's why, you know, some things aren't in the notes and they're not in the study question, and maybe some things aren't even in the book, okay? What I test on is what is, you know, basically it's like what she said. What I'm talking about in class is really what I'm testing, and I'm giving you a clue of, and again, those extra questions, you'll see some of these. I'm giving you a clue of how some of them will be boom, boom, straightforward. Others will be, you know, all the information is there, but you've got to put it together. Okay? So some question about insulin, one of the factoids might be, you know, and it is an exocrine secretion. Well, that part makes that statement false. But if it's an endocrine secretion, so if you look at insulin, now look at it. It's hydrophilic, it's protein, it can't get through the cell, it's made in rough ER, it's endocrine. So it doesn't work kind of like separate memorization. You have to start to just, you know, just start getting it enough so you start to make these associations. Okay? It's not too late to withdraw from my class. Any questions about the rest of this this chapter? God, we haven't even done. We're done. We're done with this chapter. We're not done today. We're going to do something in chapter 4 that you really it's going to be illustrating this thing called the law of mass action. But don't even look at the notes right now in the chapter. Get a fresh piece of paper out, and I'm going to do it on the board. Again, we're going to go to 7, and then we'll stop. I'm going to draw a reaction here, because I want us to see this at least once. Okay. Have we done anything with pH? No. Okay? So we skip pH in chapter 2 because it's going to apply to this part right here. So, I want to talk about pH and the pH scale. say, okay? Now, what pH is measuring, the power of hydrogen, okay, I'm not going to even put the formula because I don't even want us to know, that's representing how much hydrogen ion, H plus, is in the fluid, okay, H plus. That's what pH scale measures. And we sort of have this idea that when you have about pH of about 7, that's sort of considered neutral.
I have a quick question. Can we write that under there, or do we need space under that?
You're gonna need space on that side. I would put this on a separate side or separate piece of paper. Because I really need to explain pH and then we'll apply it to that. At least for the first time. So pH around 7 is neutral. Okay? And if you have a lower pH, pH 5, pH 3, lower than that, is that acidic or basic? Acidic. And again, a lot of people know this, and if you don't, calm down, we're telling you now, and you can look it up. So these are acids, acidic. A pH below 7 is acidic. Now the idea is, and I'll put this, even though you don't have to know, but just to put it, and we'll deal with this a little in lab, that the pH scale is the negative log of the hydrogen concentration, which means as the pH number goes down and becomes more acidic, the hydrogen concentration is increasing. The more hydrogen ion, the more acidic it is. And you've got to get that idea, okay? The more hydrogen ion, the more acidic it is. If you're higher than this, you're not acidic, you are basic, or also called alkaline. Okay? So basic pH scale. Okay? So, and again, right now, remembering acidic, basic, and the idea now is that, remember that, the more hydrogen you have, the more acidic you are. Now, we're going to see, and I'm even going to put this up here right now, that all of our proteins, etc., actually operate within a very narrow pH range of like 7.45 down to 7.35. That's what, for homeostasis now, pH homeostasis, that's the range we want. So we're going to do the up and down arrow thing. If we find out the pH is too high, we're going to have something called alkalosis. If our pH is too low, we're going to have something called acidosis. Okay? All this is going to actually apply over there. Okay? How are we going to, yes?
Is that for our bodies, or you said it most things?
Our bodies. Okay? So, yeah, we're human physiology. This is what's going to occur. We want to maintain, other organisms do this too, but we want to maintain a narrow pH for all of our enzymes, all our proteins to work. So we're going to be very careful about this, that our blood doesn't, and other fluids don't become too alkaline or too acid. We're going to have two systems that take care of this. It's going to be the respiratory system and, you know it, the kidney. Yes, the kidney. Respiratory system and the kidney. And this is the global way. I want to show you, just in general, how they're going to do this. Because the first thing you have to think about, we'll come over here, is respiration. I'm just putting RESP. But when we're breathing, what do we breathe out? CO2. Okay? And from this relationship, and we'll go into it today, and again, we're going to have to continue this on next Wednesday. The idea is that CO2 for us is basically, we're going to show this reaction, I'm just going to mention it right now. When CO2 combines with water under proper conditions, it's going to make hydrogen ions and something, and we'll write it here, called bicarbonate, which is HCO3 minus. And hydrogen ions to us mean acid. Okay? So associating CO2 with acid. Right now, that's what you without the equation, that's what I want you to think about. And when we're breathing, we're blowing out what? Which means we're blowing out acid. Whenever your body's building up too much acid, our respiratory system is going to increase to get rid of that acid. Okay? That's what I want you to start thinking about. So we're not worried about oxygen now. Don't worry. We got plenty of buffers on that too. CO2, because it's acid, can cause all kinds of problems. So our breathing is really going to be controlled by how much CO2 is in the body. So CO2, which we're breathing out, is acid. So we can even look over here. If we, for whatever reason, and we'll later deal with this, why, if we have acidosis, our respiratory system is going to respond. And the answer to this that we're going to put here, either it's going to increase respiration, breathing rate, or decrease it. If you have acidosis, what is your respiratory system going to do?
Increase.
To blow out the acid. Exactly. It will literally respond by respiration will increase. I'll put increase. Okay? It's going to increase and therefore, basically, get rid of acid. Again, it's kind of hard this way. And now the pH will come back to normal. Increase, again, I'll even put this. Yeah, even that. You're breathing out, basically, acid. So up here, if you have alkalosis, you don't have enough hydrogen. And if you look at this reaction, if you don't have enough hydrogen, don't we want more CO2 so we can make hydrogen? Okay? There's a relationship, and we'll kind of go through it probably next time more than this time, that there's a relationship here. And the idea is CO2, if we start building it up, is going to make more hydrogen. If you have alkalosis, you don't have enough hydrogen. So you need to have CO2. How are we going to get more CO2 in our body?
Hold your breath.
Right, and so decrease respiration. Okay, maybe you don't have to hold your breath. So you've got to get that CO2 acid thing. Literally, when you have alkalosis, next time, we're not going to have time this time to go into it. I almost do want to make the test go later because we really are behind. But I don't know if I can do that and make everybody happy. I'll have to see this. I could ask though, and I'll ask in a minute. But the idea is the respiratory system will decrease respiration. That way CO2 will stay, make acid, and our pH will come back down to normal.
Negative feedback.
And we'll have negative feedback for both of these. Once we're doing, our respiration rate will return to normal. This will be an immediate reaction, by the way. Okay? This is respiratory system. As soon as this happens, I wish we had more time to even finish this because it's so cool. But I might start it here. But this gets us pretty good too. As soon as this happens, the respiratory system is going to respond and do this. Either increase or decrease breathing. We restore pH homeostasis. We're cool. Who else helps us out? The kidney. 180 liters a day. Boom, it's going through. It's noting if it's acid or not. Okay? And the kidney basically decides, do I keep it in or do I get rid of it? That's what it's going to do. So, if you are acidic, your kidney is going to do what? Are we going to keep the acid in, absorb it, or are we going to excrete it into the toilet? That's what it's going to do. Of course, how it's going to do it will come later. But the kidney, as the blood's going through with the hydrogen, we have too much, is going to excrete hydrogen. Pee it out. Okay? We're going to get rid of hydrogen here. We're acidic. We're going to breathe it out and pee it out. Okay? Breathe it out and pee out. So the respiratory system is working with the kidney. Kidney's a little bit slower, but it's working over a longer period of time. If you have chronic acidosis, it's spending hours and days doing this, right? 180 liters a day, peeing out hydrogen. So, what is the kidney going to do if we don't have enough hydrogen?
Incontinence.
Basically, it's going to keep it in. You're going to pee and all that, but while it's filtering things, it's going to make sure your hydrogen stays in your body, not to the toilet. So it's going to, we're just going to put conserve hydrogen, because I'm not going to talk about how it does this. So I want you, at this point, to at least have this, okay? To have this because this is the overall pH homeostasis, how we're going to control it. Now, next time we're going to talk more about how it's going to work, and we're going to do chapter 4. It really would be nice to have this test. We're either going to have, we're going to have a choice, and everybody should be here, and I'll email it anyway, but if someone's not here, that's really not a good idea anyway.